Hello everyone. It's my pleasure to take you through our research. Integrative host microbiome modeling uncovers the implication of oral gut translocation in advanced cirrhosis. Liver cirrhosis is a major global health burden. The liver is continuously exposed to gut-derived metabolites, making it highly sensitive to microbial and metabolic changes. In cirrhosis, oral bacteria can translocate into the gut. These cross-site interactions may alter host microbiome balance. However, the functional metabolic consequences of these translocating microbes remain poorly understood. Here, we use computational approaches to investigate their metabolic functions. First, I'll highlight our main findings. Using paired oral gut metagenomics and metabolic modeling, we identified 4 key points. Oral and gut microbiomes become increasingly metabolically similar with disease severity. The species driving this convergence are predicted to produce elevated ammonia, often coupled with acetate. These patterns are supported by community-level modeling and independent validation. Finally, host modeling suggests the broader multi-organ metabolic burden. We next moved to the results. We used a reactobiome framework to capture the metabolic reaction repertoire of the microbiome, providing a functional view of microbial communities. Using data-driven clustering, we identified 3 gut and 2 oral reactotypes, both showing clear gradients with disease severity. We then quantified oral gut functional similarity using the oral gut metabolic distance. The distance decreased with disease severity, indicating progressive metabolic convergence. Given this convergence, we defined 16 translocation-associated metagenomic species pangenomes, or TMSBs. Their abundance was negatively associated with OGMD, indicating that they become more prominent as oral gut metabolic convergence increases. The genome-scale metabolic models predicted that TMSBs have significantly higher capacities for ammonia and nitric oxide production compared to healthy gut commensals. Flux variability analysis confirmed ammonia production capacity in 13 of the 16 TMSBs, and being consistent across multiple dietary conditions. In addition, a subset of TMSBs, including Veillonella atypica, showed acetate coproduction, which may contribute to exacerbating hyperammonemia in alcoholic cirrhosis. For clinical relevance, the abundance of Veillonella parvula and V. atypica in gut was positively associated severity. Network analysis further identified these species as hubs with high predicted ammonia production capacity. We further constructed community models for low and high OGMD patients. Under identical conditions, both communities achieved similar biomass. However, the low OGMD community showed higher ammonia and nitric oxide production. Perturbation analyses by introducing individual TMSBs showed 10 out of 16 TMSBs increased ammonia production in the low OGMD community, while having minimal effects in the high OGMD community. These results suggest that low OGMD patients in a more advanced dysbiotic state and are more susceptible to perturbation by translocating taxa. To validate these findings, we analyzed an independent cohort. 11 TMSBs were also detected with high prevalence. Importantly, TMSB abundance was associated with blood metabolomics linked to nitrogen burden and mitochondrial stress. We then explored the host impact using transcriptome-constrained models. Increasing ammonia uptake led to elevated flux through fatty acid oxidation, oxidative phosphorylation, and ROS detoxification in cirrhosis. Likewise, ammonia and acetate exposure was predicted to increase lactate production in the brain, and metabolic changes in muscle. These results suggest a systemic, multi-organ metabolic burden driven by translocation-associated metabolites. To summarize, our study provides computational evidence for an acetate-ammonia metabolic axis linking oral gut microbial translocation to systemic metabolic stress in advanced cirrhosis. Reactobiome profiling reveals progressive functional convergence between oral and gut microbiomes. We identify 16 translocation-associated species characterized by elevated nitrogen metabolism. Community-level modeling demonstrates amplification of these metabolic effects, while host modeling predicts multi-organ consequences across the liver, brain, and muscle. Together, these findings provide a model-based framework linking microbial translocation to metabolic dysfunction, and support the potential for microbiome-targeted interventions in cirrhosis. Thank you.